So now let's talk about the sampling distribution of the sample mean. The sample distribution of the sample mean is a probability distribution of all possible sample means for a given sample size. So what this means is, for a given sample size, the mean of all possible samples means selected from a population is going to be equal to the population mean. So what we're doing is we really have to ask the question of, how does the sample mean behave? Suppose we were to get sample after sample, finding the sample mean every time, right? So if we put these sample means into Excel and make a histogram, what would that histogram look like, right? Um, in particular, we're looking to see if that histogram has a bell-shaped or normal appearance. What we find is that there tends to be less variation in the distribution of the sample mean than what we would see in the population distribution. So when we think about the sampling distribution of X bar, or the sample mean, most of the time you're going to start in the upper right-hand corner where you have a sample of n elements selected from the population. And then you're going to use that sample data to provide the value for the sample mean. Then you use the value from X bar to make inferences about the value of the population mean, which then allows eventually for the population mean to be determined. The sampling distribution of X bar is the probability distribution of all possible values of the sample mean X bar. The expected value of X bar is going to be equal to the population mean, mu, when in this case we can determine that the point estimator is not biased. The standard deviation of X bar is the only symbol that should be new to you on this slide. And these are the values that we use to define the standard deviation of the sampling distribution of the mean. Another thing to keep in mind is that the standard deviation of the mean is also called the standard error of the mean. So when you're determining the standard deviation of X bar, you have two different formulas that you need to choose from, and it really depends on how big your sample is. So the one you choose is based on whether or not you classify your sample as being an infinite population or a finite population. Basically, a finite population is a population where the proportion of the sample size out of the population size is going to be less than or equal to 0.05. So when the population has a normal distribution, the sampling distribution of X bar is normally distributed for any sample size. In most applications, the sampling distribution of X bar can be approximated by a normal distribution whenever you have a sample size of 30 or more. However, if you anticipate that your data is highly skewed or has outliers, you might want to think about increasing the sample size to at least 50. And when you get into more complicated type models and such, sample size will obviously have to be adjusted and made larger. The sampling distribution of your sample mean can be used to provide probability information about how close that sample mean X bar is going to be to your population mean mu. So let's look at an example. In this example, we have high intensity light bulbs, and for now we're going to assume that we know what the population mean is. So we're looking at X representing the lifetime of a high intensity light bulb, where we have 20 samples, each sample containing 10 light bulbs, which is assumed to have come from a normally distributed population that we have demonstrated here. And so here we see the normally distributed curve with the population mean of being 400 hours and the population standard deviation of being 50 hours. Next, we're looking at a table that lists the means and standard deviations for our 20 samples, with a mean and standard deviation for each of the samples, for the samples combined at the bottom. And so the last four samples are shown in columns Q, R, S, and T. Two things that I want you to notice here is that the sample means are kind of hanging out around 400, which is an indication that our point estimators are fairly reasonable estimators for our population mean. And the other thing is that the sample standard deviations are all hanging around 50, And these individual standard deviations can be used as an approximation for the standard deviation of the population. As I mentioned earlier, the sample means are fairly consistent compared to the individual values, but there is much more variation in the standard deviations of the different samples, and the standard deviation of the X bar value is much lower than the standard deviation of our individual samples. Therefore, when we have a random variable, the standard deviation is going to be better approximated by taking the population standard deviation and dividing it by the square root of the sample size. So another thing to keep in mind is that sigma sub x bar is also going to be called that standard error of the mean. So let's use an example of male height with a mean of 5 foot 9 inches, standard deviation of 1/4 foot. And we're going to begin with the idea of all men and then look at a sample of 100 men. So here, this is what I'm going to call picture 1. Picture 1 is going to represent the shape of all values within the population. Here we see a mean of 5.75 feet and a standard deviation of 0.25 feet. So anytime I refer to picture 1, this is what I'm talking about. I'm talking about the values within the population. On the next slide, we're going to see a spreadsheet of 50 samples of male heights with each sample containing 100 heights. Just kind of as a side note, this actual data set was just a computer-generated example using the random function in Excel. It doesn't really come from actual measured heights. But once we made the set, each sample was selected from a normal population with a mean of 5.75 feet and a standard deviation of 0.25 feet. So the first sample is shown in column B and the fifth sample is in column F. If we look at row 101, that contains the mean of this sample, and row 102 contains the standard deviation of this sample. We would expect that the means would be hanging around 5.75 feet and that the standard deviations are in the ballpark of 0.25 feet. The first highlighted cell is the average, or the mean, of the 50 sample means in row 101. Notice that it is 5.75 to 2 decimal places, which is what we expected. Directly below this is the average of the 50 standard deviations in row 102. This is 0.25 to 2 decimal places, also as expected. The other highlighted cell is 0.025. This is the standard deviation of the 50 means in row 101, and this is 0.025 to 3 decimal places, which is what we would expect according to picture number 2. So when we look at sample averages, we get a much narrower curve. The standard deviation of this curve is the population standard deviation, which is 0.25 feet, divided by the square root of the sample size, which we were using a sample size of 100. So we're looking at 0.25 divided by 10, being the standard deviation of the X-bar curve. So what I want you to think about when you go from picture 1 to picture 2 is that you have a handkerchief hanging on top of a tennis ball. That shape would represent the distribution of the population with a normal distribution. Then when you go to pick the handkerchief up, the width of the curve becomes smaller as the height of the curve becomes taller, and your tails of your curve start to come in, which gives you the distribution of a sample, which we see here in picture number 2. So our first question is, what proportion of male heights is greater than 5.8 feet? So this picture is picture 1, since we're talking about the individual heights, and it's the same type of problem that we've done before with normal distributions. So we're going to take our values from the x-axis and use the formula for z to standardize the x values to fit them on the z-axis by using x minus x bar divided by the standard deviation. So this last expression here is basically asking, what is the probability that my x value is greater than 5.8 feet? And so when I complete this calculation, it turns into wanting to find the probability that z is greater than zero point 0.2. So now I need to look up the z value of 0.2 on the z table. So for picture 1, we're wanting to know the value of the areas shaded in red, knowing that an x value of 5.8 feet corresponds to a z value of 0.2. So when I go to my z tables, my cumulative z tables go from negative infinity up to my z-score. So since I need to find the value in red, I can look up the value, the green portion of the curve on my table, getting a p-value of 0.5793 corresponding to a z-value of 0.2. And so in order to find the value above my z-score, I'm going to subtract 0.5793 from 1, which allows for us to get that the red area is 0.4207, or basically answering the question saying that about 42% chance of randomly selecting a male who is taller than 5.8 feet. So my second question is what are the chances that an average of 100 heights is going to be greater than 5.8 feet? So this is the type of question that's going to use the taller, skinnier picture too, because we are now talking about the samples from our population. So now you're trying to determine if the sample mean is going to be larger than 5.8 feet, where X bar is the average of the heights for 100 men. So when we look at picture 2, we now see that the standard deviation is the standard error of the mean, which is calculated by dividing the population standard deviation by the square root of the sample size. The other big difference between picture 1 and picture 2 is the location of the X bar and Z axes, which are now switched from where they were in picture 1. So if you think in picture 1, you have the x-axis below and the z-axis on top with the normal curve on top. Now we have x bar on top with the z-axis below. So to move from the x-axis and the z-axis, I need to use the sample mean minus the population mean divided by the standard error of the mean, which once again is the standard deviation of the population divided by the square root of the sample size to get my z value. The standard error of the mean is going to increase as the sample size increases, because if we look, if we increase our n, that's going to increase our entire denominator, and as we increase our denominator, that's going to make my overall value smaller. So in picture number 2, the mean of the normal curve is going to be 5.75 feet. And this is the same as the mean of the population that is shown in picture number 1. The standard deviation of the normal curve is now going to be 0.025 because this is the population standard deviation from picture number 1, divided by the square root of the sample size. So using picture number 2, you can determine the z values to where you now are looking at the probability of z being greater than 2. So basically what you're doing with the x values and converting them to z values is you are now standardizing the curve by subtracting 5.75 and dividing by 0.025. One of the relationships that I would like for you to think about regarding picture 1 versus picture number 2 is that picture number 1 is going to be wider, more encompassing curve, and picture number 2 is going to be much more narrow because it's a sample. And so because it's more narrow, that's going to result in a smaller standard deviation. And when you have a smaller deviation, this is going to result in a taller curve with more of your observations concentrated towards the mean, and it's also going to result in the tails of your curve being much closer to the middle. When we look at picture 2 here, remember we found the standard deviation was 0.025, which resulted in a skinny normal curve. So our question is now asking about the probability of the average height being greater than 5.8 feet. So now we're looking for this area shaded in red. In order to answer that, we must first answer the question, what's the probability that my z-value will be greater than 2? Looking up the corresponding value for z equals 2, we see that the probability that it will be less than 2 is 0.9772. Therefore, 1 minus 0.9772 basically tells us that we really only have about a 2% chance that we have an average of 100 heights being larger than 5.8 feet. So why does the distribution mean become narrower compared to the distribution of the population? This happens because sampling moderates our tails. So when I pull my sample, I'm not capturing all of those outliers, which results in my observations falling in a more concentrated area and giving us a smaller standard deviation. Another thing to keep in mind is that if I took a sample from the area that's circled here on the screen, it would not be considered a random sample, and you would have a mean that is not representative of the population. Remember, random samples are intended to be representative of the characteristics of the population. So if picture 1 is not normal, what happens with picture number 2? So, like we said earlier, if we assume that our sample is large enough, picture 2 will still be a skinny normal curve. So if you recall, as I said earlier, if you assume that your sample points were relatively close to each other, you can use a sample size of 30 or more to still get this skinny normal curve. If you anticipate your spread or more outliers, you might want to increase your sample size up to 50.